So hello everyone. So we'll be starting with the second unit in biology, genetics and evolution. So this is going to be our first lecture. So let us begin. So this lecture will let me give you a brief outlook. So this lecture will be dealing with Mendelian inheritance mainly. Okay. So let's start this. So we need to be before we start Mendelian genetics. We need to be aware of certain terms. Okay. So let us go through those terms. Okay. So one such term is heredity. Okay. So how do you what do you mean by heredity? Okay. So heredity can be defined as some of genetic endowments. Okay, that we obtain from our parents. Endowment meaning what? Endowment meaning sort of a property. Okay, sort of a property that we get from our parents. Okay, or alternatively, we can also define it as a transmission of characteristics. Okay, structural, functional, or behavioristic, okay, from our parents, okay, to the offspring, okay. In simpler terms, you can say that characteristics like skin color, okay, like height, okay, even your manners of speech are we get from our parents, okay, our facial, the expression of our faces, our nose, the eyebrows, many characteristics that we get from our parents. Okay, so you can simply put it that it's a sum of genetic endowments that we obtain from our parents. Okay, so this science that deals with heredity is a very interesting thing that we will cover in our lectures. Okay, so what is this science called as? Okay, so this science is called as genetics. Okay, so how do you define it? Okay, so this is the science which deals with the principles and mechanisms of biological inheritance. This how are things inherited? Okay, how do we get them? So the science that deals with this is known as genetics. Okay, so students, one thing I would like to make it very clear that whenever we discuss genetics, there is a person that we cannot do without. Okay, the person who is known as father of genetics. Okay, remember this. He is Gregor John Mendel. Okay, he's there on right hand on your right lower screen. Okay, so Gregor John Mendel, he was an Austrian priest. Okay, a devout person. Okay, so he laid the initial foundations of this science of heredity. Okay, also known as genetics. Okay, I don't usually teach much history to my students, but something that's very significant here that I would like to mention to you guys is that Mendel gave his during his lifetime, Mendel was not recognized. Okay, he was very occupied with the duties towards his church. Okay, so he was very his efforts, his beautiful experiments, as we will see later on in these lectures, whatever he did, decades of his work was not recognized in his lifetime. Okay, it was only after, I think, 2 or 3 decades after his death that scientists such as botanists such as De Vries, Schermark, and Correns, okay, proved that yes, Mendel was indeed correct with all of his experiments. Okay, so the reason I'm telling this to you is that students should be, it should inspire students that sometimes if you put efforts, sometimes if you put efforts into your work, it may be that in that instant it may not get recognized, but as time passes, your efforts will bear fruit, and the truth will ultimately prevail. Okay, so this is exactly what happened in Mendel. Okay, several decades after his death, the genetics, took, the boom of genetics came into science. Okay, the term genetics was coined, several decades after Mendel's death by Bateson. Okay, so let us move on. So why is Mendel so famous in genetics? Okay, what did he do? What were his experiments? Okay, so let us take a look. Okay, so Mendel's experimental material, was pea plant. Okay, very famous plant, in the context of genetics. Okay, मटर जिसको हम कहते हैं in Hindi. Okay, so the garden pea plant was a very good choice for Mendel. But why? Okay, what are the reasons for it? Okay, first and foremost, okay, it was true breeding. Okay, meaning the offspring, had the same characteristics generation after generation. Say, for example, you wanted to follow any trait, the trait remained consistent throughout several generations. There was no change, there was no variation in trait from one generation to next. Okay, this is very important when you're studying genetics. Okay, if the change is naturally occurring, how will you study genetics? Okay, so something has to be remain constant. Okay, moving on, it has contrasting traits. Okay, see, when you are studying genetics, how will you study it? You will study it, if you have a tall guy and a short guy. Okay, if you have a if you have a black person and you have a white person, so you can study the traits easily, because you need to have contrasting traits. Okay, so that is exactly what is the case with pea plant. Okay, pea plant has flowers, purple, white. It has seeds, a round, wrinkle. Okay, there are many characters that Mendel selected. There were 7 of them, which we will study in the next slides. Okay, but the point is that the experimental material should have contrasting traits. Point number 3 is that the pea plant was capable of self pollination. Why is that? As we will study in the next slides. Okay, during his experiments that Mendel did with the pea plant, he needed self pollination. He needed self pollination the pea plant. Okay, so in this, the flower of the pea plant is special, that it is protected from foreign pollen. Okay, so sort of self fertilization is favored in the pea plant. Are you getting my point? Okay. So the shape of the pea flower itself makes it protected from pollen, as a result of which Mendel was easily able to do self pollination in the pea plant. Moving on, point number 4, it grew quickly. Okay. See, Mendel couldn't have done genetic experiments with an elephant. Okay, which needed so much food, which needed so much space. Okay, so it grew quickly and required very, very small space. In fact, if I'm not mistaken, Mendel grew these pea plants in the small garden, backyard garden of his monastery. Okay, so moving on, the plant is small, yes, but the flowers are large enough. Okay, large enough to be handled by us. Okay, why do we need to handle flowers of pea plant? See, sometimes we need to remove the anthers. Sometimes we want to remove the pistils. So they must be large enough so you could work with them. So you could work with them easily. Okay. Flowers are bisexual, meaning flower has both the male and the female part with it. This is the reason there is self pollination in pea plant, because the flowers are bisexual. Okay. So I would suggest you to study the structure of flowers if you guys are not aware with this. Okay. So what is a pistil? What is a anther? Study that. Okay. Female and male parts of the plant. The last point, that chances of contamination foreign pollen were very, very little in case of pea plant. This is a very important point. Okay, it may be asked in exams as well. Why? Because the self pollination in the pea plant occurs in the bud condition itself. Okay, even in the bud condition, the pollination is done, and the game is over. Okay, so there's no concept of cross pollination occurring in a pea plant. Okay, when the pollination has been done even in the bud condition. So this is a sort of benefit that the pea plant has. Okay, to prevent it from cross pollination. Okay, so this is why the experiment, the garden pea plant was a very, very excellent choice for Mendel. Okay, so moving on, regarding experiments, what were they? Okay, so see, broadly, see, the details of Mendel's experiments I'll be discussing in the next slide, but this is just a broad overview of how did Mendel approach his experiment. Okay, what was the broad steps that Mendel did during his experiments? Okay, what did he exactly do? We will study it in the next slides as I discuss Mendelian inheritance further on. Okay, but if you want to see that how did Mendel approach his experiments, you must be aware of 4 steps that Mendel did. Okay, first was selection of parents. Okay, ये experiment किसके साथ करेगा? Okay, what did he need to do the experiments? Okay, so next came hybridization. Okay, and then self pollination, and then calculations. Okay, we'll be discussing these individual points. Don't worry, we'll be discussing these individual points in detail. Okay, so first comes selection. So Mendel picked the pea plant. Okay, so yes, fine, but what were the characters that he was looking for in the pea plant? Mendel initially picked many characters, 34 of them, but at last he settled for 7 characters. But remember, with contrasting traits. Okay, remember this. Okay, every character had to have contrasting traits. So let me make this clear with a slide I have kept. Okay, so have a look at this. Okay, so Mendel selected 7 characters in the pea plant, which he used, and each character had 2 contrasting traits. Okay, say for example, you have over here. Okay, say for example, you have over here the flower color. Okay, you have over here. Let me just move on to this pointer options. Okay, let me keep laser option over here. Okay, so so this is a character. Okay, flower color is a character, pea shape is a character, pea color is a character, pea pod color is a character, pod shape, plant height, and flower position. So these all were characters that he picked. Okay, so these are traits. See, these are traits: purple, white, contrasting traits. Okay, this is what P shape. The P, when you extract, जब आप मटर निकालते हो, from दाने निकालते हो, how are they? They can be round, they can be wrinkled. Flowers, they can be purple, they can be white. P color, again, color. P color is a character. Okay, trait, they can be yellow, they can be pod color, they can be green, they can be yellow. Okay, similarly, pod shape, they can be inflated or constricted. Okay, plant height, it can be tall, it can be short. Okay, very simple, straightforward slide that I kept for you guys to explain it in very easy manner. So flower position, axial or terminal. You understand axial and terminal? See, these flowers are all in the terminus, meaning at the end. So these are at the axis. Okay, they just branch off, just towards the left or right. Okay, so these were the characters that Mendel selected, during his experiments. Okay, then came hybridization. Okay, hybridization means, how did he mixed two plants with each other. Okay, so this involved what? So this involved cross pollination. Okay, so this involved cross pollination. So how did he make this cross pollination feasible? Because sir, you just said before some slides, because there was, what, pea plants are always favored to do what, self pollination. Okay, so how did he do this hybridization? Okay, first and foremost, contrasting traits were selected. Okay, here it is. Okay, so contrasting traits were selected, meaning, so when he did the hybridization, there was purple, pea plant with purple flower and pea plant with white flower. Or if it's pod shape, it was round and wrinkled or constricted. Okay, so contrasting traits were selected for two plants. Okay, so one plant was made a male. How? By removing the pistils. Another was made by a female by removing what? Anthers. Okay, that's easy. Okay, I hope you guys know what is a stigma is, what is a anthers are. Okay, you just have a look at the parts of flower for me. Okay, so this is how he created a male and a female plant, and then he did what? Hybridization, meaning what? Cross pollination. He dusted off pollen grains from one flower to the next. Let me show you a slide. Okay, next slide. Okay, so here it is. Okay, so this is a purple flower, and this is a white flower, contrasting traits. Okay, so one was made a male, another was made a female, and then there was what by the way, this process is known as what? This process is known as emasculation. Okay, in some the stamens were removed, those behaved as female parent, and in some the pistils were removed, those behaved as what? The male parent. Okay, so this is how male and females were created. Otherwise, remember one thing. Otherwise, the flowers of pea plant are what? Bisexual. Both sexes are there present in the single flower. But you had to make male flowers and female flowers by emasculation. Then came bagging. Okay, bagging means what? To protect them. Okay, so that there isn't any unwanted cross pollination. Okay, so this is how we do it. And then what? Dusting. Dusting means to brush the stigma of the emasculated flowers with what? With the pollens of the other sex. Okay. So tagging involved what? To label them. Experiment kin plants pe hua hai. Okay, and this is how he did it. Okay. There are crosses. Mendel did many crosses: monohybrid, dihybrid, trihybrid. This we will discuss in the next slides as we move further in these experiments. Okay. By the way, you have to remember that when they did this was a male plant, this was a female plant, and then they did cross pollination. Okay, so then there were seeds. Okay, they had to again then there were seeds, pea seeds from this. Then they had to again grow this. Okay, then came the F1 generation. Okay, meaning first filial generation. This was the first generation that was obtained after what? Cross pollination. Okay, it's not just enough to cross them. You have to cross them. Yes, then the seed has to be what? It has to grow in the soil, and then the whole new plant has to come in the next season. So Mendel was very patient with his experiments. Remember that. In science, you require what? Patience, students. Okay. In science, you require patience. Only then can experiments be carried out. Okay. So this is how he did his experiments. Okay. Third came self-pollination. Yes, whenever Mendel first was the selection, second was the cross-pollination, third was what? Self-pollination of the hybrid. Okay, so in this, in this Mendel needed to do any bagging or emasculation, because self pollination is the default way the bee plant what the flowers reproduce. Okay, the flowers pollinate, okay, by self pollination. But remember one thing, the seeds of the plants that were obtained by cross pollination, okay, in the previous slide, they are known as F1 plants, and the seeds that are obtained after self pollination are known as what, F2 plants. Don't worry, I will explain all these terms in detail in the next slides. This is just a broader view that how did Mendel did his experiment. Okay, I'll be discussing each of these terms separately. I have kept these separate slides. Okay, so moving on, calculations. Okay, calculations. Okay, so how did this is shown over here. You can see over here. These are the initial parents. This was cross pollination. Okay, then came self pollination. Remember, this is the offsprings after cross pollination were F1 over here. Okay, and then these were self pollinated, and then we have F2. Remember this. Okay, so parents, cross pollination, F1, then self pollination, then F2. Okay, so you see, if you the character that Mendel took over here was what, seed color. Okay, this is yellow. Okay, this is yellow, and this is green. Okay, so see, when he pollinated the yellow with the green, all the offsprings were yellow. Okay, all the offsprings had what, yellow colored seeds. Okay, there was no green. Okay, why? This is due to dominance. I'll tell you later on. Okay, but the reason I'm showing you this slide is because Mendel had to count each and every plant. Okay, Mendel counted each and every plant in this case. Okay, so this is this is the F2 generation. Okay, see, when these were again self pollinated, the trait appeared. Okay, see, the trait appeared over here. Okay, but when these guys were some in the F2 generation, some when they were self fertilized again, these were sort of homozygotes. There was these were pure breeds. Okay, these had some of the green seeds, and this was the these are also the pure lines. Okay, so these were also the pure lines. Okay, so these are the calculations that Mendel did. Okay, whatever plants he obtained the F2 generation. Okay, they were ratioed out. Okay, they were ratioed out. Okay, so 2/4 were found to be true breeding. Okay, so these are what, these are true breeding. Okay, these are true breeding, and 2/4 50% were what, hybrids. They gave tall as well as dwarf. These. Okay, this we will discuss in the next slide. Okay, but these are just the initial calculations that I wanted to discuss with you. Okay, before we move on to the next slides. Okay, so genetic terminology. Okay, so there are some basic genetic terminologies before we move on to the actual Mendelian inheritance. There are experiments, many things. Okay, there are a few terms that you guys must be aware of. Okay, so what are they? Genes, alleles. Okay, so how do we define? Yes, gene. Okay, so as you can see, if this is a cell, this is a nucleus. You take out its chromosome. There is a DNA. Okay, and on the DNA we have a gene over here. So how do you define a gene? Okay, so this is a beautiful little context that I have given you. Okay, so gene is an inherited factor that determines a trait. Okay, so as you saw in the previous slides, there were color, flower color, color, seed shape, plant height. Who controls that? Who controls that actually? Okay, who controls that? Who determines the trait? It is these genes that determine it. Okay. So this term was initially coined by William Johansen, okay, a Danish geneticist. Okay, so this is how we define a gene. Sir, what is an allele? आपने सुना होगा, sir, allele किसको कहते हैं? Okay, so alleles can be defined as one or two more alternative forms of a gene. Okay, see, whenever we have a gene, we must have one gene from our father and one gene from our mother. Okay, say for example, a trait that I've shown over here is hair color. Okay, yes, for hair color you have one gene, one paternal gene from your father and one gene from your mother. You guys know this because we have we have homologous chromosomes. Okay, we have one set, 23 chromosomes we get from our mother and 23 chromosomes we get from our father. Every chromosome has a pair or its replica from the other parent. Okay. So whenever we have a gene, we must have its counter allelic part. So alleles of a particular gene will be found at a specific locus on a chromosome known as locus. Okay, say for example over here. Okay, moving on. Character. This I touched. We were lucky enough to touch it in the previous slides. That character. Okay, how can you define a character? Okay, character can be defined as a distinct, morphological or physiological feature. Okay, say for example, we don't need to take the examples from Mendel's experiment. We can simply say, character can be defined as what, color of your hair. Okay, height of a person. Okay, similarly in case of Mendel, it can be shown as stem height. Kitni hai? Is it tall or is it dwarf? Flower color, is it purple or is it white? Okay, shape of the seed, वो wrinkled है या round है. Okay, so this is a character. Okay, character means what feature you are picking. Okay, वो feature कैसा है, that comes under trait. Okay, whatever feature you are picking, say for example height, that is a character. How much is it? Is it tall or short? That comes under what? Trait. Okay, so is it tall or dwarf? If you pick up hair, is it curly or straight? Okay, that is a trait. Okay, so please remember, students, what is a character and what is a trait. Okay. So moving on. Okay, so if you take from a previous slide, we discussed that allele. Okay, if there is an allele, okay, see, alleles can be homozygous or alleles can be heterozygous. Okay, so homozygous means, if you talk about homozygous, an individual possessing two of the same alleles at a locus. If these two alleles are same, okay, these are homozygous. Okay, if these two alleles are different, okay, like this, okay, if they have two different alleles, they are called as what? Heterozygous. Yes, why not? Because you have two sets, one from your father and one from your mother. If they are similar, they are homozygotes. Okay, if they are dissimilar, they are heterozygous. Now you will be asking me the question, sir, why is there a capital A over here and why is a a small a over there? Okay, this we will discuss in the next slide when we discuss dominant and recessive. So have a look at this slide. Okay, so have a look at this slide. So see, we have two alleles for a gene. Okay, over here. So one is one I have represented by a capital A and another I have represented by a small. So why have you done this? The allele that is represented by a capital letter is the dominant allele, or you can say the dominant gene. How can you define it? It refers to an allele or a phenotype that is expressed in the presence of an alternative allele. See, there are always alternative alleles over here, but the thing that is expressed, that is the dominant one. See, you have two alleles. How do you detect a dominant one? The thing that is expressed and which shows its effects. Okay, even the term dominant means which expresses itself. Okay, that is the dominant gene. Okay, so in this case, okay, it is a dominant gene. Obviously, dominant gene will show its effects whether it's homozygous, ये दोनों same है, और इसके साथ कोई दूसरा है recessive. It will show its effect. That's why I've written over here. It expresses effects both in homozygous and heterozygous condition. Okay. So what about this? ये छोटा a क्या होता है? Okay. These are very, you can say them, they're sort of a weaker gene. Okay, they will express themselves only in homozygous condition. They बेचारे थोड़े डरपोक होते हैं. They will only express themselves in homozygous condition. Okay, similar to what people who are not very brave are. Okay, they express themselves only under limited condition. So similar is the case with recessive gene. Okay, so they express themselves only in homozygous condition. Okay, like this, when there is a small a and small a in this case. Okay, it refers to an allele or phenotype that is expressed only in homozygotes. Okay, when both the alleles are similar, only then will a recessive gene be expressed. Okay, are you getting my point? Only when both the alleles are same, जब दोनों small a है, then it will be expressed. In this case, when it has a neighbor जो काफी dominant है, in this case the recessive gene will not express. That's why it's called as recessive. Okay. So dominant genes are expressed both in homozygous and heterozygous conditions, and recessive genes are expressed only in what? Homozygous conditions. Okay, they are not expressed in heterozygotes. Okay, when capital A or small a hai, as in case of this will be expressed and not. ये बेचारे डरपोक होते. So remember this. This is very, very important points over here. Okay? So moving on, genotype and phenotype. How do you define a genotype and a phenotype? Okay. So see, I have given a nice little slide over here. So these are the alleles, okay, as I shown you. Okay. So this is a heterozygote. This is a heterozygote. Okay. If you cross them, okay, we will get four combinations. Okay, we will get homozygous A, heterozygous capital A, small a, and homozygous A. So these are genotypes. Okay. But how will their phenotype look like? Okay. So have a look at this. Okay, this is the flower color I have taken. Okay, so P is a allele for what? Flower color. Okay, so see, sir, how can you know what what trait is dominant and what is recessive. I'll ask you the question in this slide. How do you know which trait is dominant and which trait is recessive? Simple answer is, sir, look at the heterozygous genotype. Ye wala. So look at the heterozygous genotype. Okay, the color that is expressing in that is the dominant one. Okay, the color that is expressing in the heterozygote is the actual dominant color. Okay, purple. Okay, as you can see, I told you this in the previous slide as well. Okay, so purple color is the dominant color in case of in this flower, say for example. How, sir? How can you tell us that? Because the purple color is expressed in heterozygous condition. Is expressed in homozygous as well as heterozygous conditions. Whereas the color white is recessive. See, it's neither expressed here, but it's only expressed when it is in the what homozygous condition, as I told you previously as well. Okay, so one important thing that I would like to tell you guys is genotype doesn't change, most of the time. Okay, but phenotype changes to a certain extent in environment and age. Okay, phenotype changes. Okay, so phenotype can be discussed, can be defined as appearance of manifestation of a character. Okay, if there is a character, how is it manifested? Genotypes are simply when you look at the allele. Okay, so this is the genotype. Okay, and this is the phenotype. Okay, so this is a nice little image that I have for you. Okay, so these are the genotypes and these are the phenotypes. Phenotype is what you see physically. Okay, and genotype is what you analyze when you're looking at the chromosomes or alleles. Okay, you can remember it this way. Okay, okay students, now we move on to Mendelism. Okay, now that we are done with the basic concepts of genetics, genetic terminologies, and how did Mendel construct his experiments. Okay, now we move on to the actual principles of inheritance that were laid by Mendel. Okay, so what do we call them? We call them as Mendel's principles of inheritance, or simply Mendelism. Okay, they were formulated by Gregor John Mendel. Okay, so how many principles of inheritance are there? Okay, there are 5 principles of inheritance as formulated by Mendel. Okay, and 2 of them are called as law of segregation and law of independence, which are the significant one. Okay, we'll be giving much of our attention to these 2 laws. Okay, we'll study them in detail. We will discuss we will discuss terminologies associated with them. Okay, so be patient and grab your pen and paper and keep taking on notes with this lecture. Okay, so in total there were 5 basic principles of Mendelism. Okay, so let us discuss the first 3. Let us discuss the first one. Okay, so first one is called as the principle of unit character. Okay, so it's a pretty simple and a straightforward law. It says that each organism, whether you take a pea plant, whether you take a human being, whether you take an animal, any organism, has a large number of characters. Okay, the word character. What does this word? Let me just go to the pointer options. Just a minute. Highlighter. Yeah. Okay. So if I were to tell you that, what does this term mean? Okay, what does this term mean? What do you mean by a character? Okay, as I discussed with you in the previous slide, that what can you call as a character? So you can call height as a character. Whether tall or dwarf, that's a different thing. So you can call skin color as a character. Okay, whether a dark or light, that's a different matter. Okay, so you can take eye color as a character. Whether blue or black or brown, that's a different thing. Okay, so remember what is a character and remember what is a trait. Okay, we already discussed in the previous slides, but if you didn't pay attention over here, this is a beautiful opportunity for you guys to clear your concepts up. Okay, you must be able to differentiate between a character and a trait. Okay, eye color, as shown in the screen over here. Okay, as shown in your screen over here, eye color is a character. Okay, whether blue, brown or black eyes, that's a trait. Okay, height is a character. Okay, whether tall or dwarf, those are units. Okay, so students, you must know the difference between a character and a trait. So moving on, this principle states that each organism has a large number of characters. Okay, skin color, eye. Oh, in case of peas, you might be wondering, sir, what is the character in terms of peas? I think we discussed this in the previous lecture. Let me take you back over there. In pea plant, 7 main characters were chosen by Mendel. Let me just take you guys back over here. Okay, so these were the characters that were chosen by Mendel. Okay, so this is flower color, pea shape, pea color. Okay, pod color. See, when we are studying Mendelism, when we are studying genetics, we are mostly focused on the pea plant. Okay, so if you want, we will be discussing many characters in our next experiments, but all of them will be more or less focused on these 7 traits. Okay? So you must remember these 7 traits. Okay, memorize them. Okay, memorize them. What are the 7 traits in Mendelism? Okay, so these are the 7 traits of Mendelism. Okay, sorry, 7 characters that Mendel used to study genetics in the pea plant. Okay, so remember these 7 characters, and these characters are what we will be discussing in the slide. Okay, so let us go back to our slide of Mendelism. Okay, so it says that since each organism has a large number of characters, each character behaves as a unit. Okay, so one unit we call as one character. Moving on, the second principle of Mendelism, what is that? Okay, it's called as a principle of paired factors. Okay, it simply states that it's pretty straightforward. Again, law. Okay, it says that each character is represented in an individual by 2 unit factors. Okay, so what are those 2 unit factors called? They are called as alleles. Again, we discussed both of these terms, character, allele, in the previous slides when I told you the genetic terminology. If you guys don't remember it, you can go back to the video and you can check it up over there. Okay, so see, each character, say for example, you take a character, say for example, of what, flower color. Okay, so flower color will be represented in a pea plant by 2 unit factors. What is that? Alleles. Okay, those are alleles. Okay, so sir, why the paired factors? Why 2 alleles? Because if I take your attention over here, because one allele we get from our father and other allele we get from our mother. Are you getting my point? So that is why, whatever character you are studying, whether it's flower color, whether it's height, whether it's your shape of a nose, shape of your lips, okay, yes, it is represented by a unit, as said in the first law, first principle. But each character, it will be represented individual by 2 unit factors known as alleles. Okay, so alleles, what are simply alleles? Okay, they are 2 basic variants of a gene at a given locus. See, this is the locus. This is the locus. Locus meaning a specific position in a chromosome. Okay? There'll always be 2 alleles. Okay. Sir, what do you mean by homozygous and heterozygous? Again, we discussed it previously, but let us discuss it again over here. Okay. So you have 2 alleles. Okay. You have 2 alleles. Beshak, okay. But they can be same or they can be different. Okay. See, in this case, they are what? In this case, they are same. Okay? You can call them as what? Homozygous. Okay. So here I've mentioned it. Okay, you can call them as homozygous. In this case, see, in this case, the alleles are different. Okay, okay. One I have written in capital, and another I have written with a small letter. What does this mean? I will explain it in the next slide when we will take up the third principle of Mendelism. Okay, but they're different. The alleles are different. Okay, I have shown this capital by black, and I've shown this small by white. So in this case, the alleles are different. We will call them as what? We will call them as heterozygous organisms. Again, okay, you see, I know this is your first lecture of Mendelism, so I'm intentionally repeating many things so as to make the concepts very clear. Okay, so again, so this is homozygous when the alleles will be same. Okay, in this case, or you can say that. See, I have shown a very good sentence over here. Okay, in case of homozygous, the alleles have the same trait of the character. Okay, in case of homozygous, the alleles have the same trait of a character. Okay, say for example, you are taking height. Okay, say for example, you are taking height. This will be tall. This will be tall. Both of them will be tall. Okay, if you are taking a heterozygous, see, in heterozygous conditions, okay, they have different traits of the same character. Okay, character वही है, character height है. Okay, but in this case it's tall, and in this case it's dwarf. Okay, in this case it's tall, and in this case it's dwarf. Okay, so in homozygous organisms there are similar alleles having the same trait. Remember this. Okay, I'll just focus on this. Let me go back to pen. Okay, in homozygous, okay, they have the same trait. Okay, in heterozygous they have different traits. Okay, over here. Okay, one will be tall and another will be dwarf, whereas in homozygous it will be tall, or it will be dwarf. It will be dwarf, dwarf. Okay. Now, of course, the question will be, sir, why have you just let me just erase this. Let me just erase a bit. Okay. So now the obvious question will be, just a minute. Yeah. Okay. So now the obvious question will be, sir, why have you mentioned, why have you shown some alleles as capital and why have you shown some alleles as, in a small letter? Okay. So the ones that I have shown in capital are the dominant traits, and the ones that I have shown in small are the recessive traits. Remember, traits. Okay. Not characters. Character is the height, and trait is the whether it's tall or dwarf. Okay. So, sir, what do you mean by this? This will be explained in the next slide. Okay, when we take the third principle of Mendelism, called as principle of dominance. Okay, this is what I was telling you guys. Okay, this is what I was telling you guys. Okay, so uh, have a look at this. Okay, so whenever if you take the third principle into consideration, out of the two factors or alleles, okay, as I told you, there will always be two factors or alleles representing different traits of a character. Okay, this law states that out of the two factors or alleles representing different traits of a character, only one expresses itself. Remember this. Okay, only one expresses itself. Okay, say for example, if the case in this case, okay, in this case, if we have two factors or alleles, okay, of different traits. Remember, okay, listen to my words very carefully. Out of the two factors or alleles representing, see, the word is over here, representing different traits. When the traits are different, say for example, there is one trait with a capital P, another trait with a small b. Okay, out of the two factors or alleles representing different traits of a character, only one expresses itself. Sir, who expresses itself? Okay, it will always be the dominant one. Ye wala. It will always be that's why it's called as dominant. It shows its dominance. Okay, it shows its dominance. That's what I have shown in the definition over here. If you watch this, the factor allele which expresses itself, okay, which expresses itself, the word is very clear, okay, which expresses itself in a hybrid is called a dominant factor. Okay, the other allele, okay, the other allele, jo bechara, it's sort of a weak guy. It's very, it's not able to, so as to say, it's not able to fight. Okay, it doesn't dominate. The other factor allele which is unable to show its effect, okay, which is unable to show its effect in the presence of an alternative allele. Okay, जब ये अकेला होता है बेचारा, at that time it will show its effect. Okay, in this case, ये वाला. Are you guys following me? Okay, in this case, when it will have similar recessive alleles over there, it will show its effect. Yes, okay, it will show its effect. That is what we call as homozygous for a recessive allele. See, so this is, in this case, when it will be homozygous for the recessive allele, at that time the recessive allele will show its effect. But when the recessive allele will be in the presence of its alternative allele, it will not show its effect. That's why it's called as a recessive factor. Okay, so that's why. Okay, so see, when can you know whether an allele is dominant, or which is a dominant or which is a recessive? When you have conditions known as what? As such as heterozygosity. Okay. So see, so in this case, recessive allele showed its effect. Okay. So this is pod color. Okay, yellow and green. Okay. So see, in this case, it is recessive, right? But it's showing its effect. How come? Okay, because it's in a homozygous recessive condition. Okay. And in this case, this is homozygous dominant. Of course, it will show its dominant. May problem nahi hota hai. Dominant will always show its effect. Okay, regardless whether it's homo or heterozygous. Okay, but see, when we do a cross of homozygous and heterozygous, I'll discuss these crosses later on. Okay, these are gametes. Okay, these are gametes Y and this Y. Okay, see, this is a heterozygote, right? So this is a heterozygote. Okay, there is Y allele, capital Y allele, as well as there is small y allele. But असर किसका हम देख रहे हैं? Which is showing its effect? Is yellow showing its dominancy or green is showing its dominancy? Yellow is showing its dominancy even in the presence of what? The recessive allele. That is why it's dominant. Okay, I hope I have made this clear. Okay, so in homozygous everything is jolly good. Okay, in homozygous this is yellow, in homozygous this is green. But what if when they are crossed? Okay, when we have a heterozygote, who shows this effect? The trait that shows its effect is the dominant trait. Are you guys following me? Okay. So there are always dominant traits and there are always recessive traits. Okay, there is yellow color and there is green color. Okay, so anytime you want to see anytime if I were to ask you the question like this in this cross, if I were to ask you the question like this in this cross, which is the dominant trait? How will you answer my question? Sir, we have to look at the what? We have to look at the heterozygous individual. Okay, जो heterozygous individual में हो रहा है, जो trait heterozygous जो trait heterozygous individual में express हो रहा है, that is the dominant factor. Okay, जो trait heterozygous individual में express हो रहा है, that is the dominant factor. Okay, let me just want to this one here. Let me just take you to pen. Just a minute. Okay, so जो trait heterozygous individual में जो display हो रहा है, that is the dominant. So you can clearly see heterozygote में it's yellow, that means yellow is the dominant one. Okay, see one thing I want to make clear, this is in case of pod color. Okay, ऐसे ही काफी सारे dominant recessive traits है. Sir, you will ask me the question, sir, what are they? What are other dominant and recessive traits? Let me take you back. Okay, let me just show you that character chart again. Very important slide that I've kept in this. Okay, so see, okay, you can clearly see. Okay, जो ये वाले है, okay, जो ये वाले traits है, they are dominant, and जो ये वाले है, okay, जो ये वाले है, they are what? Okay, they are recessive ones. Okay, जो हम अभी example the example जो मैं दिखा रहा था अभी, that was what? वो ये थी पी कलर. ओके, मटर का कलर कैसा है? ओके, वेदर इट्स येलो, यहां पर ये येलो है, और इट इज ग्रीन. ओके, तो इफ यू आर वंडरिंग, ठीक है, ये पी कलर में है, येलो डोमिनेंट है और ग्रीन रिसेसिव है. सर व्हाट अबाउट पी शेप? उसमें डोमिनेंट कौन है? उसमें राउंड डोमिनेंट है और रिंकल्ड रिसेसिव है. ऐसे ही इफ यू सी फ्लावर कलर, सर उसमें डोमिनेंट कौन है? व्हाट इज द डोमिनेंट वन इन दिस? पर्पल कलर इज द डोमिनेंट वन and white is a recessive one. Again, if you see pod color, so pod color में कौन dominant है? Remember this, pod color is different and p color is different. Okay, p means अंदर वाला जो दाना होता है and pod color means the outer covering. Okay, pod color में dominant कौन है? Pod color में green dominant है and yellow recessive है. Again, if you move on further, pod shape में inflated dominant है and constricted recessive है. Meaning what? अगर हम say for example inflated pod shape और constricted pod shape वाले pea plant को क्रॉस करेंगे, एक्सप्रेस कौन होगा? Inflated one will dominate in heterozygous condition. Okay, so let us move on to the remember this slide. Okay, students, okay, let me just rub it off. Okay, my suggestion to you guys would be that remember this very this is a very important point. Okay, so let me just take you again over here. Okay, let's go back to the slide. Okay, so this is the principle of dominance. Okay, so remember that so this is the case. So this is the case of what? This is the case of homozygous dominance. This is the case of homozygous recessive, ये वाला, and this is the case of heterozygous. Okay, इसमें कौन एक्सप्रेस होगा? सर जो डोमिनेंट होगा, that is expressed, and recessive is not expressed. Okay, let's move back to the next slide. Okay, see ये जो तीन थे, principle of unit factors, okay, principle of unit factors, principle of paired factors, and principle of dominance, they were ये पहले तीन laws है. These are the first three laws of Mendelism. Okay, इसके बाद now comes the interesting part. Okay, now comes the interesting part known as what? Laws. Okay, law of segregation and law of independent assortment. Very easy laws. Just pay attention, guys. Okay, so now moving on, we have principle of law of segregation. Okay, what does it say? Okay, what does principle of law of segregation say? It states that the two factors of a character. Okay, the two factors of a character. Okay, say for example, height is a character. Okay, इसमें दो factors होते हैं ये वाले. Okay, ये वाले दो फैक्टर्स होते हैं, okay? The two factors of a character present in an individual do not get mixed up, okay? Okay, they don't get mixed up, they remain distinct, meaning जो हमें जो दो alleles हमारे पास होते हैं, we have two alleles, okay? They don't get mixed up, okay? They segregate, they segregate properly into from each other at the time of what? Gametogenesis. This is gametogenesis. ये, okay? When these are when this is what? This is gametogenesis. Okay, जब भी gametes बनते हैं, you guys know this, that gametes में जो हमारे पास the two alleles that we have in in a diploid cell, you guys know when gametes are formed, gametes are haploid. Okay, gametes don't have both the alleles. Alleles separate out when gametogenesis occur in a process known as what? A meiosis. Okay, in a process known as meiosis, two alleles are given to the two cells in a process known as meiosis. Okay, so this is a normal cell, this is the diploid cell, okay, and this is the haploid cell. Okay, haploid cell will always have one allele. Okay, haploid cell will always have one set of genes, one allele. That is what this law says. Okay, that two factors, जो ये दो factors होते हैं, that these two factors in individual do not get mixed up, okay, but remain distinct and they separate from each other during gametogenesis, so that जो gamete होता है, gamete के पास खाली क्या एक ही T रहता है. Let me just move on to pen or laser. Okay, so gamete has only what? Gamete has only one T. Okay, इसके जो यहाँ पर it had two alleles T and T. This is homozygous. Okay, remember this is homozygous dominant. Okay, जो ये दो alleles है, they separate into into two different alleles in during gametogenesis. Okay, so you can say that gamete carries only one factor simply. Okay, so that this law states that this gamete, when it is formed from a diploid cell, it carries only one factor of the character. Okay, and is always pure. It has to be pure. Why? Gametes homozygous or heterozygous नहीं होते. Okay, gametes are always, they have single alleles, so इसमें homozygous or heterozygous की बात ही नहीं है. Okay, they are always pure. That's why this law says. Okay, that's why this law is also called as what? Let me just go on to the highlighter. Okay, see guys, that is why this law is known as principle of purity of gametes. Okay, a nice little sentence over there. That's why this law is known as purity of gametes. Gametes are always pure. Pure means there is no concept of homozygous heterozygous over there, because इनके पास एक ही alleles होता है, तो of course they must have only one set. Okay? So this is also known as principle of purity of gametes or the first law of Mendel. Okay, so how can we explain this? This can be explained with the help of a monohybrid cross. Okay, now we'll ask you the question, sir. What is a monohybrid cross? Okay, in monohybrid cross, okay, we are only studying one character. The cross which you are seeing in your screen has only one character. कौन सा character है? Right. Only one character is being studied in this cross, so that's why we call it a monohybrid cross. अगर दो character होंगे तो dihybrid. अगर तीन characters we are studying, then it is trihybrid. We will discuss dihybrid cross shortly. Okay, so this is a diploid cell. Whenever gametogenesis occurs, it carries only one allele. Okay, so the gamete carries only one factor, and that's why it's also always pure, and it's also known as the law of purity of gametes. So let's take this cross further on. Okay, so what is this? This is splitting itself into gametes. Okay, ये भी gametes बना रहा है ऐसे और ऐसे. Okay, so now if I ask you the question, what is happening in this step? Okay, what is happening in this step? F1 generation कैसे बनी? I'll ask you this question. Okay, that how is this F1 generation formed? Is it formed by cross pollination or it is formed by self pollination? If you guys listen to my previous slides where I was discussing the basic constructs of how did Mendel do his experiments, Mendel F1 generations are synthesized how? They are synthesized in a cross pollination manner, meaning okay, let me just go back to pen. I have to use pen in this because I have to show you what I'm explaining. Okay, meaning, okay, one gamete should one gamete should be male and another gamete should be female. There must be cross pollination, and again If I ask you the question that how did Mendel make this male, and how did Mendel make this female? See, they are very, very beautiful questions. Pay attention, guys. Otherwise, now everybody can understand crosses and everybody can understand monohybrid dihybrid cross. But I need you to consider this. How is the FN generation formed? ये कैसे बना? This ये वाला. Okay, how is this formed? Okay, how is this tall formed? Now you will ask me the question, sir, why is it tall? Because tall is a dominant gene, it will show its effect. Okay, but how is this formed? By cross pollination. Sir, cross pollination कैसे हुई? See, that's why I'll just take you to the previous slide because these are your initial lectures. You guys must be knowing this. Okay, so see, hybridization. Okay, see, this was the previous slide that I showed you. Mendel experimentation involved 4 steps. Okay, selection of parents, hybridization, self pollination, calculation. The step that I am showing you, ये कौन सी step है? ये वाली step, ये वाली step है, hybridization. So how did you do hybridization? Okay, this is how he did hybridization. Okay, one flower was made to act as a male, another flower was made to act as a female. Okay, एक से pistil remove किया गया और दूसरे से what? Okay, stamens remove किया गया. Okay, so Mendel made male and female plants, and then F1 generation बनी. See, ये काफी सारे बार क्वेश्चंस भी आता है that F1 how are F1 generations formed? F1 generations are formed by cross fertilization. Okay, remember this. Okay, or cross pollination. Okay, remember these points. Okay, how is F1 generation? These are that's why I discussed these basic experiments in the start. Okay, let's move back to our slide. Okay, so this is how the F1 generation is formed. Why? इसको F1 क्यों कहते हैं? This one is first filial generation. Okay, ये पहली generation है. Okay, so this is the tall. Let me just go back to this pointer. Okay, so this is the tall plant. Okay, now what happens? Okay, now what happens? Let me just go this thing here. अब see, now we have the tall plant already. How is इसका genotype कैसा है? It looks something like this. T and T. It is a heterozygote. Okay, I hope you guys are following me. Okay, so it is a heterozygote. Okay, so it is a heterozygote. Okay, so gametes will always be what? So gametes will always be pure. Okay, so let us move on to the next slide. Okay, lastly, the principle or law of independent assortment. Okay, so what does it say? The principle of law of independent assortment also is called as second law of Mendelism. Okay, principle of law of segregation we called as first law of Mendelism, and principle of law of independent assortment we call as second law of Mendelism. Okay, so what does it state? Okay, it says that factors or alleles of different characters. See, now the word is what? Let me just go over here. Now the word is different character. Okay, meaning, see, you should automatically understand that, sir, if you are taking different characters, so cross kaisa hona chahiye? How will the cross look like? It will be a dihybrid. Okay, when you're taking more than one characters, it must involve a dihybrid cross. Okay, so what does it say? Alleles of different characters, okay, sort separately or independent of one another at the time of gametogenesis and do not interfere with one another. Okay, do not influence one another. Sir, आप कौन से characters की बात कर रहे हैं? Say for example, if I take two characters like seed color and seed texture, okay, are the two characters which is chosen over here. Yeah, sir, if you do a cross between you, they will always remain as such. They will not influence each other. Are you getting my point? They will never interfere with each other. That is what this law basically says. The law of independent assortment says that factors or alleles of different characters assort separately. Okay, they are very independent of each other. Seed color, जो seed color का alleles है, they will never influence the alleles of seed texture. Are you getting my point? They will assort independently. See, say for example, if I draw a diagram over here, just a minute. Yeah, okay. Okay, so this is a chromosome. This is another chromosome. Okay, say for example seed color का allele यहाँ पर है. Okay, okay, और seed texture का allele यहाँ पर है. दो alleles. Okay, we always have two alleles for a different. Okay, say for example ये seed color के दो alleles है और ये seed texture के दो alleles. Okay, दो alleles है. So they will always they will always separate out independently. They will not influence each other. Okay, meaning this set will assort independently of this set, or characters not interfere with each other, meaning this will not influence this. Okay, this will never influence. Okay, so how do you understand it? By a dihybrid cross. Sir, वो क्या होता है? This is the dihybrid cross. Okay, सर आपने कौन कौन से कैरेक्टर इसमें लिए? Okay, what are the characters that you're taking? Okay, I'm taking what? Okay, I'm taking over here. Okay, uh, you know uh, this P color. Okay, yellow color. Okay, and P shape. Okay, okay, I'll take the P color. Okay, yellow. Okay, and P shape. Okay, P color कैसा हो सकता है? P color येलो हो सकता है और ग्रीन भी हो सकता है. ओके, ऐसे ही पी शेप राउंड भी हो सकता है और रिंकल्ड भी हो सकता है. ओके, राउंड भी होता है हो सकता है और रिंकल्ड भी हो सकता है. ओके, इफ यू टॉक अबाउट पी कलर, ओके, इसमें डोमिनेंट कौन है और रिसेसिव कौन है? सर येलो इज द डोमिनेंट वन एंड ओके ग्रीन इज द रिसेसिव वन. ओके, पी शेप में the round is the dominant one, okay, and wrinkled is the recessive. Okay, okay. So sir, how can you tell us this? Sir, आपको कैसे पता चला that yellow is the dominant one? In terms of P color, yellow dominant है green के ऊपर, and in terms of P shape, round dominant है wrinkled के ऊपर. Because when we do a cross of this, remember, when we do a cross of this, again students, if I ask you the question that ये कौन सा cross है? This is cross pollination. Okay, this is not selfing, remember. Okay, this is cross pollination. How is it done? एक plant को male बनाया जाता है, एक flower को male बनाया जाता है और दूसरे flower को what is made a female. Okay, I hope I'm drawing right. Yeah. Okay, another is made a female. So ये कैसे होती है? This I told you in the previous slides. Emasculation, bagging, tagging. Okay, that is how we make male and female plants. See, there is a reason we have to do it. We have to make a male plant and we have to make a female plant because the flowers of pea plant are bisexual. Remember, guys. Remember this very very important point. You have to make them male and female. Otherwise, they are what? They are bisexual. Okay. So this is the F1 generation. What do we get? Okay, इसमें कैपिटल Y का एक एलील. ओके, इन दो में से एक एलील. ओके, एंड R का एक एलील एंड स्मॉल r का एक एलील. So how will it look like? It will always look yellow and round. Sir, why not, sir? Green और wrinkled के alleles भी है because yellow and round is dominant over green and wrinkled. Are you getting my point? Okay. Sir, what do you mean by this? We're not getting this. Let me take you back to this slide. See, okay. Yellow P color is dominant over green P color. Are you getting my point? Round P shape is dominant over what? Wrinkled P shape. This is what I'm showing you in my slide. Remember this. Remember this important slide. Remember this whole slide. Okay? Remember this whole. This is a very very important slide. Okay, guys. So let us move back to our slide. Okay. Okay. So now we have. Sorry, there's a lot of junk over there. Let us remove this. Okay. Yeah. Okay. So this is what? This is a this is a heterozygote for टू कैरेक्टर, एक कैरेक्टर क्या है, पी कलर, ओके, जिसमें येलो और ग्रीन है, एंड अनदर इज पी शेप, ओके ओके, जिसमें राउंड एंड रिंकल्ड है. नाउ व्हाट डू, वी डू द सेल्फिंग, ओके. F1 जनरेशन क्रॉस पॉलिनेशन से बनता है और F2 जनरेशन सेल्फिंग से बनता है. सेल्फिंग मीन सेल्फ पॉलिनेशन, ओके. सो दिस इज दी, ओके, ये इसके गैमेट्स है, ओके, ये इसके गैमेट्स है, ओके. अगेन सी, नाउ यू आस्क मी द क्वेश्चन, सर गैमेट्स में आपने अभी बोला गैमेट्स आर ऑलवेज प्योर, इसमें Y और R क्यों? सी, व्हेनेवर गैमेट्स आर, yes, gametes do, you know what, they are pure in nature, they segregate out, but they will always have, they will always, they need to have, alleles of both the what, of both the characters, okay. ये एलील, ये वाला एलील पी कलर का है, और ये, सॉरी नॉट एलील, ये वाला जीन पी कलर का है, और ये वाला दिस जीन व्हाट, पी 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 शेप का है, ओके. सो दिस इज ये ए गैमेट है, वाई आर. सी, gametes will always have the genes for both the, since यहाँ पर हम दो कैरेक्टर्स स्टडी कर रहे हैं, गैमेट्स में दोनों कैरेक्टर होने चाहिए, ओके. गैमेट्स में दोनों कैरेक्टर होने चाहिए, आइदर दोनों डोमिनेंट, ओके ओके, इन दिस केस आइदर दोनों डोमिनेंट, आइदर दोनों रिसेसिव, आइदर एक डोमिनेंट एक रिसेसिव, और इसमें एक रिसेसिव एक डोमिनेंट, okay. This is how gametes are made, okay. Gametes should have all the genes, okay. Let me just wrap this, okay. So remember these points, students, okay. So remember these points, a minute. So remember these points that इसमें दो characters, you are studying two characters in this, P color and P shape. Remember students that whenever you are making gametes, make sure gametes में दोनों कैरेक्टर रिप्रेजेंट हुए होने चाहिए, ओके. एक गैमेट में दोनों कैरेक्टर्स रिप्रेजेंट हुए होने चाहिए, ओके ओके. इसमें इसमें पी कलर का भी होना चाहिए और पी शेप का भी होना चाहिए. वेदर होमोजाइगस डोमिनेंट, वेदर होमोजाइगस रिसेसिव और हेटेरोजाइगस, दैट्स अ डिफरेंट थिंग, बट दोनों कैरेक्टर्स रिप्रेजेंट होने चाहिए, ओके ओके. सो ऐसे ही these are the male gametes, okay. These are the female gametes, meaning what, the ova, okay okay. So we have male gametes and female gametes, we do the selfing, okay. So cross this, okay. What will you get? Capital Y, okay. Capital Y, capital R, okay. How will it look like? Yellow round, okay. इसमें capital Y, capital R, small r, ये. How will this look like? Yellow and round. So round क्यों? Because a round shape is dominant over what? Wrinkled shape. इसमें, okay, just cross, just take a pen and a paper and just draw this. Practice this, students, okay. Practice this. You need to practice this with a pen and a paper, okay. So practice this. इसमें क्या आएगा? Capital Y, small y, and capital R. Okay, this is what is showing over here. Okay, again this will be yellow round. इसमें capital Y, small y, and capital R, small r. Okay, yeah, this is yellow round. Okay, similarly you can do this for let me just rub this. These are simple. Let me show you some. Let me show you where you can, यहाँ पर आप, where will you get confused? Okay, let us show this. Yeah, let us show the cross between this and this. Okay, इसमें क्या आएगा? Okay, let us show the cross between these type of gametes. Okay, gametes हर किसी टाइप के बन सकते हैं, हर किसी टाइप का क्रॉस हो सकता है. Okay, इसमें क्या बनेगा? small y और capital R. Okay, so this will not be yellow. Remember, this will be green. Why? Because the green listen to my words very carefully, sir. Because the green P color is recessive in nature, it will always express itself when it will have what? Homozygous recessive condition, meaning small y, small, or capital R, capital R. This will be green and round. Okay, this will be green and round. Green is a recessive trait, and round is a dominant trait. Okay, green in color, round in shape. Okay, let's take this. Let's take this. Let's take a cross between this. Take a cross between. What will you guys? What will you guys get? Okay, small y, capital Y. Okay, it's over here, and small r, small r. Okay, so this will be yellow and wrinkled. Again. See, wrinkled. It will only be in that condition when there is what? Homozygous recessive. So this is homozygous recessive. It will be wrinkled. See, this is also homozygous recessive. Small r, small r. This will be recessive. Okay, this will Schwartz effect. Okay, green will only be expressed when there is a homozygous recessive condition. Let me just wrap it up again. See, this is a very important slide, so that's why I'm taking as much of time as possible. Okay, so see. Okay, so where is green expressed? Green is expressed over here. Homozygous recessive conditions. Homozygous recessive conditions. Homozygous recessive conditions, because I told you recessive, you can say the recessive allele is a weaker allele. Okay, it will always डरपोक होता है. जब ये अकेले होगा ना, दोनों recessive alleles होंगे तब ये आके express होगा. Okay, और ये भी है कौन? Homozygous recessive condition. So green will always be expressed when it will have the homozygous recessive condition. Okay, ये green किसकी बात कर रहा हूं मैं? P color. Okay, sir, what about this P shape wrinkled? Wrinkled कब express होंगे sir? Wrinkled ये रहे. Small r, small r. Homozygous recessive conditions. Homozygous recessive conditions and homozygous recessive conditions. Okay, so it will be expressed in this case. Okay, sir, कोई केस है जब दोनों रिसेसिव होंगे? या दैट्स अ वेरी स्पेशल केस. वो ये है, वो एक ही होगा. क्या? स्मॉल y and small r, small r. Remember, small meaning recessive allele and capital meaning dominant allele. जब दोनों present होंगे, dominant allele will be expressed. Okay, so see, something he has shown over here is the इसको हम F1 generation बोलते हैं, which is formed after cross pollination, and this is F2, which is formed after selfing, when we self pollinate them. Okay, self fertilize them, and then so this is the F2 generation. Okay, he has shown that इसमें when we have F2 generation, इसमें we will have 4 types of plants. Okay, देखो, pay attention guys. Okay, round yellow. Okay, round yellow, ऐसे वाले. Okay, एक तो येलो होंगे और एक गोल होंगे. कितने हैं ये? 1 2 3 4 5 6 7 8 and 9. See, you have 9 rounded yellow. Sir, what about wrinkled yellow? Okay, जो येलो होंगे मगर wrinkle होंगे, ये वाला है, and this and this. We have 3. Okay, so that's why there is 3. Okay, round green, 1 2 और 3. We have wrinkled green, 1, ये वाला. Are you guys following me? Okay, so the ratio will come out what? The ratio will come out to be 9:3:1. Okay, but remember students, one thing I want to tell you, just let me just rub this all. One thing I want to tell you guys is that sir, ये ratio कौन सा है, by the way? See, ये ratio sir, ये ratio कौन सा है? Pointer options, highlight. Okay, sir, ये ratio कौन सा है? This is the phenotypic ratio. Okay, this is the phenotypic ratio. Okay, because see, आपने count किए, now आपने count किए 3, आपने count किए 3, आपने count किए 3. ये किस basis पर आपने किए? आपने genotype देखा कि आपने phenotype देखा? Again, if you don't know genotype by phenotype, see my previous slides over there. Okay, so what do you see? Sir, आपने खाली physical characteristics देखे. Okay, so this is the phenotypic ratio. Remember this. Okay, this is the phenotypic ratio where you just simply see what. See, I will just show you the pen. Remember this. Remember this point. Remember this very very important point. Okay, so this is the phenotypic ratio. जहां पर हम खाली ये देखते हैं ये. Remember this statement, guys. ये. Okay, just the what? Just the physical aspect. ये. Okay. So this is the phenotypic ratio. Okay, मोटा मोटा हम देखते हैं जो वैल्यू जैसे ये राउंड है, wrinkled है, कलर क्या है, बस. This is the phenotypic ratio. Okay, there is another ratio known as the genotypic ratio. This I will leave to you guys. Okay, this is an assignment that I'm keeping for you guys. Let me just wrap this all up. Just a minute. Okay, second is called as what? Pointer options. Second is called as the genotypic ratio. Okay, so genotype क्या होता है? This is the genotype. Okay, this is the genotype. Okay, ये वाला. Are you guys following me? Okay, so this is the genotype. Okay, so genotypic ratio is different and phenotypic ratio is different. So I'll leave this to you guys. Okay, so what is phenotypic ratio? तो मैंने बता दिया that it's 9:3:3:1 in case of a dihybrid cross. Remember this. In case of monohybrid cross, it is what? In case of monohybrid cross, it is 3:1. Okay, it is 3:1. Okay, but in case of dihybrid cross, the phenotypic ratio is 9:3:3:1. I'll keep one thing to you guys. Tell me what is the genotypic ratio in case of a dihybrid cross, like this one, that you're seeing in the on your screen. Tell me what is the genotypic ratio of all of them. Very very important point. These things are usually asked in competitive exams. Since you guys are preparing for competitive exam, these things are asked. Okay, if the phenotypic ratio in case of a dihybrid cross is 9:3:3:1, then what is the genotypic ratio in case of this? Very easy, just calculate it. Okay. So what is the genotypic ratio in case of this? I'll just hint it. Let me just hint it. Okay, let me just hint it over here. Let me just wrap this first. Okay. जीनोटिपिक रेशियो. सर हम जीनोटिपिक रेशियो कैसे कैलकुलेट करें? यू हैव टू सी सेम कौन है. देखो इन जीनोटिपिक रेशियो. ओके, सी इन फिनोटिपिक रेशियो ये दोनों सेम है. लिसन टू माय वर्ड्स वेरी केयरफुली गाइस. इट्स अ ब्यूटीफुल पॉइंट. आई विल जस्ट हिंट इट यू. व्हेन यू कैलकुलेट फिनोटिपिक रेशियो, ये और ये सेम है. सॉरी, या इन केस ऑफ फिनोटिपिक रेशियो ये दो सेम है. फाइन, दोनों येलो राउंड है. बट इन केस ऑफ जीनोटिपिक रेशियो ये सेम नहीं है. ओके, बिकॉज ये दोनों येलो तो है, येलो होमोजाइगस दोनों है, बट ये राउंड होमोजाइगस है और ये राउंड हेटेरोजाइगस है. ओके, इन फॉर कैलकुलेटिंग जीनोटिपिक रेशियो ये डिफरेंट होंगे. सर जीनोटिपिक रेशियो में सेम कौन कौन होंगे फिर? सी गाइस, पे अटेंशन गाइस. ओके, पे अटेंशन ओवर हियर. सर जीनोटिपिक रेशियो में सेम कौन होंगे? ये और ये सेम है. आर यू गेटिंग माय पॉइंट गाइस? इन जब आप जीनोटिपिक रेशियो निकालोगे, ओके, जैसे आपने फिनोटिपिक में 9 3 3 और 1 लिए, जीनोटिपिक रेशियो में अब ये सेम है. ओके, बिकॉज ये येलो होमोजाइगस है, राउंड हेटेरोजाइगस है. फॉलो मी. ये येलो होमोजाइगस है और राउंड हेटेरोजाइगस है. ये सेम है इन जीनोटिपिक रेशियो, ये सेम है. आर यू गेटिंग माय पॉइंट गाइस? ओके, सिमिलरली जीनोटिपिक रेशियो में ये दो सेम है. ओके, व्हाई? बिकॉज ये हेटेरोजाइगस येलो है, होमोजाइगस राउंड है. हेटेरोजाइगस येलो है, होमोजाइगस राउंड है. आर यू गाइस फॉलोइंग मी? ओके, वेरी वेरी इंपॉर्टेंट पॉइंट. ऐसे ही ये दो सेम है, ये दो सेम है इन जीनोटिपिक रेशियो. ओके, व्हाई? बिकॉज ये ग्रीन होमोजाइगस है और राउंड हेटेरोजाइगस. ओके, दिस इज हाउ यू कैलकुलेट द यूनिटिपिक रेशियो. ये सेम है. ओके, तो वेरी वेरी नाइस. अगेन देखो, अगेन ये भी सेम है. इन जीनोटिपिक रेशियो में ये दो सेम है. सी, ओके, येलो हेटेरोजाइगस एंड राउंड heterozygous. This is how you calculate the genotypic ratio. Okay, so interesting assignment for you guys. Okay, so make sure guys you tell me that what is the genotypic ratio in case of a dihybrid cross. Okay, okay. So in case of a monohybrid cross, the phenotypic ratio is 3:1, and genotypic ratio have 1:2:1. Whereas in case of dihybrid cross, the phenotypic ratio is 9:3:1. And tell me what is the genotypic ratio. Okay, okay. So see you guys in the next lecture. Okay, the next lecture will be covering non-Mendelian inheritance. Okay, it's an interesting one. Where we'll be discussing complete dominance, co-dominance, many things. Okay, so make sure you guys whatever I have taught you in this lecture, make sure you revise it, take notes, study this again from your books. Okay, okay, so I'll be see you guys in the next lecture. Okay, so have a great day.